Hello everyone, my name is Moshada Das. I have completed my masters in botany from Vishwabharati University. Then I did a computer course on DOFP. Now I am currently working as a subject matter expert at Chegg India. So I am a biology teacher and I will tell you about cell a bit today. So let's begin. So cell is a fundamental unit of our life. So it is a self-replicating structure itself. So first cells were observed was Robert Hooke in 1665. Um, he discovered cell from slice of cork. Some organisms consist of a unicellular organism. So here is structure of a cell. You can see there is there are many more internal structure of a cell like nucleus, mitochondrion, Golgi body, DNA, endoplasmic reticulum, lysosome, cytoplasm, cell membrane, centriole, peroxisome, vacuole, ribosome, Golgi complex. Many more internal structure are present in our cell. So cell theory is proposed by Matthias Sladen and Theodore Swan in 1839. They said that all living things are made of cells. Cells are the smallest working unit of all living things. All cells come from pre-existing cells through cell division. So they described cell theory actually. So there are here are some constitution of cell. Constitution means বাংলাতে বলে উপাদান তো there are so we can see the substance like protoplasm is present in cell. Protoplasm is composed of 70 to 80% of water which is present in cell. So there are major structure in the cell membrane, cytoplasm, its organelles and nucleus. So there are many more structure in the cell membrane, cytoplasm and its organelles and nucleus. We will see in next slide more. um, so here we can see cell membrane. So cell membrane is a thin, pliable, elastic outermost structure which envelops the cell. Means it guards the cell. It consists of bilipid layer with embedded proteins that are uh, um, differentiated by two proteins. Um, Functions of cell membrane are forms outermost boundary of cell organelles, which I have said previously. Digest it takes in food and excretes waste products. Non-polar molecules like gases, O2, CO2, NO2. Polar molecules, chemical, physical properties. So links adjacent cells together by junctional complexes to form tissues, and there are insulating properties. It acts as a dielectric material of a charged condenser. Thus cell membrane have very high insulating value. Now we will see cytoplasm and its organelles. So cytoplasm is a liquid form of structure, and organelles float in this structure. So it is inside the cell. So let's see what So this is cytoplasm. Cytoplasm is a thick gel-like semi-transparent fluid that is found in both plant and animal cell. The constituent parts of cytoplasm are cytosol, cell organelles, cytoplasmic inclusions, bounded by plasma membrane and contains many organelles in eukaryotic cell containing then cytosol, the aqueous part of the cytoplasm outside all of the organelles, also contains its own distinctive proteins. So there is 70% of cell volume. There are present many gelatinous substances like cytoskeleton, filaments, organic molecules, salt and water. Now come to organelles. These organelles are present in cytoplasm, which uh, floats in cytoplasm. These are mitochondria, lysosomes, peroxisome, endoplasmic reticulum, Golgi apparatus, vacuole. So each organelle is bounded by a lipid membrane. Now we will see mitochondria. So mitochondria were first observed by Kollikar in 1950. So it is a granular structure in striated muscles. It is called the powerhouse of the cell. There are many more structures in mitochondria, which are inner membrane, outer membrane, Christie matrix. So the length is 5 to 12 micrometer and diameter is 0.5 to 1 micrometer. So components of mitochondria are outer membrane, inner membrane, intermediate space, Christie matrix, which I have told in previous slide. So these membranes are made up of phospholipids and proteins. Now we will see the outer membranes and interior of mitochondria. In outermost membrane there are many integral membrane proteins present called porins. There are many enzymes. Interior matrix of mitochondria is present. There is inner membrane. Inner membrane contains ATPase and other enzymes concerned with synthesis and metabolism of ATP, contains enzymes of electron transport chain. The ultimate purpose of these mechanisms is oxidative phosphorylation and synthesis of ATP. Mitochondria had some protein synthesis. Functions of mitochondria is power generating units of cells, important to maintain proper concentration of calcium ions within the various compartments of the cell, energy transaction through respiration, responsible for thermogenesis. If you need the PPT, then comment below in my video so that I can provide you the PPT. Endoplasmic reticulum. So network of tubular and flat fascicular structures of cytoplasm. An extensive network of closed flattened membrane bounded sacs called cisternae. Space inside the tubules. There are differentiation between smooth endoplasmic reticulum and rough endoplasmic reticulum. So in ACR ribosomes absent, RDR contains ribosomes, site of synthesis of lipid and steroid hormones, site of protein synthesis, processing and packaging, mainly present in lipid forming function of ER: synthesis of proteins, protein segregation, unsaturation of fatty acid, muscle contraction. ER is commonly known as sarcoplasmic reticulum in muscle fibers. If you need any guidance or any online classes, then let me know in the comment section so that I can arrange accordingly. So now we will learn Golgi bodies. So Golgi bodies is a collection of membrane enclosed sacs composed of 4 or more stacked layers of thin, flat enclosed vessels lying near the site of nucleus. Consist of multiple uh, function is wrapping, packaging department of the cell. You just have to go through this PPT very carefully and very attentively, and then you can learn the structure of the cell and the function uh, very good. Then we know lysosomes. So uh, so this is a very long video that I tried my best to interactive to uh, the lesson. So lysosome, its diameter is 250 to 750 nanometer. Uh, Contains oxidases that produces H2O2, catalyzes degrades hydrogen peroxide to yield water and oxygen. Proteins are directed to peroxisome by a unique signal sequence with the help of protein chaperones peroxisome. So here we can see the function H2O2 metabolism and detoxification, helps in photorespiration in plants, biosynthesis of lipids, cholesterol, glycol are synthesized in animals, synthesis of bile acids in liver, synthesis of plasmalogens, myelin sheath. So that's it for the function. Now cytoskeleton. So cytoskeleton is a system of fibers that not only maintains the structure of the cell but also permit it to change shape and move. The cytoskeleton is made up of primarily of microtubules, intermediate filaments, microfilaments along with protein and that anchor tie them together. There are many more structures in cytoskeleton function. They are involved in movement of the chromosomes, cell movement processes that move secretion granules in the cell, movement of proteins within the cell membrane, uh, and many more functions. This is a vast syllabus, I have uh, concized it in a very in a short PPT. So you have to read both NCERT, and if you in West Bengal board, you have to read both Bengali book and Jivan Vikan and the Sahika. The nucleus contains chromatin, RNAs, and nuclear proteins move freely in aqueous solution. Nucleus has an integral structure that organizes the genetic material and localizes nuclear functions. A loosely organized matrix of clear lamina These lamins serve as sites of chromatin attachment and organize other proteins into functional nuclear bodies. Chromatin within the nucleus is organized into large loops of DNA, and specific regions of these loops are bound to the lamin matrix by the lamin binding proteins in the chromatin. Now we proceed with nuclear envelope. So, complex structure consisting of two nuclear membranes and underlying nuclear lamina and nuclear pore complexes. There are two concentric membranes called the inner and outer nuclear membranes. The outer membrane is continuous with the endoplasmic reticulum, so the space between the inner and outer uh, function acts as a form of digestive system of the cell because enzymes present in it can digest essentially or macromolecules engulf worn out components of the cell in which they are located, engulf exogenous substances like bacteria and degrade them. It is called societal wax. Now paroxysm. So paroxysm, the diameter of paroxysm is 0.5 micrometer. So a lipid bilayer membrane surrounds which regulates water entrance and exits the paroxysm. Um, Urate oxidase crystalline core structure is similar to that of lysosomes but with a different composition. So here are microtubules, intermediate filaments and microfilaments. The definitions and the features are given uh, in this. You have to go through this all to clear your doubts and also the circle of your knowledge. Nuclear membrane is permeable onto small nonpolar molecules. Underlying the inner nuclear membrane is the nuclear lamina, a fibrous network that provides structural support to the nucleus. So, um, now function serves both as the repository of genetic information and as the safe control center. The presence of nucleus thus allows gene expression to be regulated by post-transcriptional mechanisms such as alternative splicing. The nuclear envelope provides novel opportunities for the control of gene expression at the level. Now, here is summary compartments and major functions. You can go through this to a concise idea about cell compartments and the major functions of the organelles which are given in this slide. So that's it for today. Thank you for your time to see my first presentation video. Also keep watching my channel. I will upload many more topic video of biology, and I will be uploading many videos of biology, so stay tuned.